Hello everyone, my name is Tarnima Omara and I am a master's student in the Hygiene and Zoonosis Department at the Faculty of Veterinary Medicine, Mansoura University. My focus is on antimicrobial resistance and I have been actively working with whole genome sequencing and more recently exploring metagenomics approaches as a part of our Labs One Health research efforts. Today, and by the end of this video, I promise you'll understand in a super easy and beginner-friendly way how to go from raw metagenomic data till visualizing and comparing microbial communities. We'll break everything down step by step using very clear explanations and practical tools to guide you through the whole process. So let's get started. Today, we'll be discussing taxonomic profiling and visualization of metagenomic data. First of all, I want you to know that if you are completely new to concepts like taxonomic profiling, taxonomic assignment, or even metagenomic data, this is completely fine. You don't have to worry as we are going to explain everything from scratch. And regarding our objectives for today's tutorial, you can just ignore them for now. The only thing that I need you to know is that we are going to start with raw metagenomic data, the DNA of all organisms in our sample, and end by gaining the ability to visualize and compare the microbial communities. And instead of jumping straight into technical terms, let me take you on a little thought experiment. You have just to close your eyes and imagine. Imagine you're dropped into a forest you have never seen before. You know this forest is full of life, but you can't see the creatures directly. There is nothing to help you. No binoculars, no field guide, just scattered clues. You can see shadows, you can see rustling leaves, paw prints, and maybe broken branches. All of these refer to the fact that there are living organisms in this forest. But what exact species are found? What exact counts are there? You have no idea. That's exactly what it's like looking at raw metagenomic data. So today we'll embark on an expedition into this mysterious forest. Not with scanners, not with maps, not with guidebooks, but with our powerful tools. We can use Kraken 2, Metafilan, and Corona, and then we'll be able to uncover the hidden species in our sample through using these powerful tools. Now that we are deep into this mysterious forest, let's talk about the clues we are actually working with. In metagenomics, we don't find feathers, we don't find rustling leaves or branches or footprints like we have said in the forest metaphor. Instead, we find DNA. That's our clue. So what's metagenomic data? What is the definition of metagenomic data? It is the DNA from all organisms in a sample. It is a mix of genetic material from all the living organisms in a sample. Whether that sample is soil, water, or even a human stool sample. So based on this, metagenomics allows us to study microbial communities directly in their natural environment without the need for culturing. So you don't have to stand for hours in your lab, do the isolation things and bacterial culturing. Instead, we can just use metagenomics, okay? And this is so important because most microbes cannot be easily cultured. Yet they play a huge role in ecosystems. Now that we understand what metagenomic data represents, it's time to build a solid foundation. So let's take a brief pause from our expedition journey, and break down some of the core scientific concepts that guide how we classify and analyze our metagenomic data. Firstly, the microbiome. What is the microbiome? Microbiome refers to all the microorganisms living in a specific environment, plus the conditions they live in, and everything they do. It's like a whole ecosystem, not just a collection of microbes. So, as it's written here, microbiome is a community of microorganisms in a habitat like gut, like soil, or water in a pond, for example. And this includes their DNA, environment, and activities. Like I've said before, it is a whole ecosystem, not just a collection of microbes. Shifting towards taxonomy, think of taxonomy as a biological filling system. Every organism is classified into levels, domain, kingdom, phylum, all the way down to species, based on shared traits or shared characteristics, especially their DNA. Okay? And surely the system helps us identify or organize life on Earth. Just to make it simple, imagine you are at a big family reunion, okay? You start by recognizing broad groups like everyone with the same last name. Then you narrow it down: cousins, siblings, parents, grandparents, and so on. Biologists do the same thing with organisms, okay? They start big like domain or kingdom, same as we start big with broad groups. And then they zoom in step by step until they reach the individual species. Like I have said in the family reunion example, you start by broad groups and then you narrow it down: cousins, siblings, parents, grandparents. This is the same thing, okay? So, in a nutshell, taxonomy is how we organize life and understand who's related to whom. And this is, of course, related to like when we are talking about living organisms. How do we actually figure out who is in a sample? That's where taxonomic assignment comes in. We compare DNA sequences, like DNA sequences from our sample, to databases of known sequences, just to identify which exact microbes are found in our sample. Some tools do this by comparing DNA to genomes, like DNA to DNA, as Kraken tool. Other tools compare DNA to proteins, and some tools look for unique marker genes. If this unique marker gene is found, then the specific organism that contains this unique marker gene is found, which is logical. And one of the examples on the tools that look for or search for unique marker genes is Metaphylla. Shifting towards taxonomic profiling, taxonomic profiling isn't just about identifying who is there, but it also gives us information about how many of each species is found. Different tools use different strategies for this, like some tools align our reads to full genomes, and this method is accurate but like resource heavy, and other tools other tools like break DNA into k-mers, which is way faster but less accurate or less precise. We can summarize this slide and say the most important points. First of all, I want you to know that microbiome is like the bacteria in your gut, like the microbes in the ocean water, and so on. About taxonomy, we can give an example like E. coli. E. coli belong to domain. Bacteria, for example, genus Escherichia and species coli. Then taxonomic assignment comes in. For example, we can say Kraken2 tool compares your sequences to known genomes just to identify the microbes in your sample. This method is the taxonomic assignment that Kraken2 tool use. About profiling, in a nutshell, profiling helps answer two questions. The first one is what microbes are there, and the second one is how many microbes are there. Now that we have covered the key scientific concepts, let's take a closer look at the real environment we'll be exploring. We are not going to explore the forest like we have said in the beginning of our video, but instead we are going to explore a hidden oasis in the middle of the Mexican desert. This is our expedition site. It's called Cuatro Ciénegas, I guess. I hope I am pronouncing it the right way, but that's our forest for today. This place is home to some of the most unique microbes on Earth. living in an ancient pond ecosystem there. So, scientists ran a full-scale experiment. They added nutrients to one pond and left the other pond as a control one to see how the microbial community would respond. Like they just want to check or to see if changing the ecosystem would affect the creatures there or not. They want to see whether changing the ecosystem would make some creatures appear, vanish, or it will not affect these living creatures at all. So we'll explore two zones. Like we have two samples. The first sample is JC1A. It's our control sample. It is a sample from the untouched natural pond. And the second sample is JB4D. This is a sample from the fertilized pond rich in nutrients. Okay? These samples are like the footprints and traces left behind by creatures in two parts of the forest. One is the part disturbed, and the other part is the untouched one, the control thing or the control zone. Okay, and our job is to uncover who was there, how many, and why. So now we are ready to start our scanning journey. So in this journey through the mysterious microbial forest, these are essential tools. These tools in front of your eyes are the essential tools we will put in our backpack. Each one had a special job to help us find, count, and understand the tiny creatures hiding in our samples. Let's get started with these tools. The first tool is Kraken 2. Kraken 2, I can call it the creature scanner. It's like a fast radar that sweeps the forest. About its rule in summary is that it quickly checks all the DNA in our sample to see which microbes might be there. It has some advantages and some disadvantages. About its advantages, it's like super fast and gives us a wide overview, like a radar picking up movement. But the negative point is that Kraken 2 might not always be that accurate or that precise. You can think of it like studying. If you are studying, for example, a chapter rapidly, you're not that perfect, you're not doing your job accurately, and so on. So Kraken is fast, but not accurate. Not always of the accuracy we need, or of the highest accuracy. Then comes Bracken 2. Honestly, I forgot to add Bracken 2 to the tools list, but Bracken 2 is the smart field guide. It's like a species logbook with built-in counters. It takes the list from Kraken, so Kraken might, if we add it, we will add it here after Kraken. Okay? So Kraken tool takes the list from Kraken and helps you get better counts. Like if I am in the first, if we are talking about the first example, Kraken would tell us what kind of organism, what kind of species are there. Then Bracken tool comes to tell us the exact counts, like how many wolves, how many rabbits, and so on. Then comes Metaphylan. Metaphylan tool, I can call it the genetic magnifier. It's like a magnifying glass that spots tiny special clues. In summary, Metaflan looks only for very unique DNA signs to tell exactly which species are there. Since it searches for unique DNA markers, it is like super accurate. On the opposite side, it is like much slower and only finds a species it already knows. So, Metaphylan is the exact opposite to Kraken. Kraken 2 is super fast but not that accurate, and Metaphylan is super accurate but slower, but much slower. Then shifting towards Corona, our magical map. It's a glowing circular map you can zoom and spin. By the way, the first slide in this video has a figure. This figure is Corona output. Can we show it? This is Corona output. Corona turns our results into a beautiful interactive chart, and we can explore from big groups like bacteria to specific species like, for example, E. coli. And we can zoom in and out. It's great for seeing and sharing what we found. Then, our last tool for today is Bavian. Bavian is our detectives board. It's a board that helps us compare different areas. It shows the differences between our samples. Like if we have two samples, it shows the difference between our two samples to help us see which microbes appeared, which disappeared, and which grew in numbers. In summary, each of these tools reveals a different clue. When we use them together, we get the full picture of our microbial mystery. Before going through explanation of each tool separately, we will be learning how to add data to Galaxy. I've just opened Galaxy Europe, and I recommend you to use the same Galaxy server, Galaxy Europe, or the same Galaxy instance instead of using different one like, for example, Galaxy Australia. And then we will open this C Galaxy training material. We will click on it. It will open the learning pathways. We will scroll down till reaching this Metagenomics data processing and analysis for microbiome. We'll click on it. This will open, and then we will search for our tutorial. It is the taxonomic profiling and visualization of metagenomic data. We'll click on it, and then we will scroll down to preparation of Galaxy and data. This part. You can just click on this. This part includes creating a new history and then renaming the history. We will do this now. We just click the plus button here, and then we rename the history from the edit button, typing the new name, and then save. Just like this. We will create new history from this plus button, and then we will name the history from the edit. We will name it anything like taxonomic profiling, for example, and then we will save. So we have created a new history and named it also. We will move to the tutorial once again, and then we will get to the part of importing datasets. How to import our data from Zenodo. These are the links for our samples. We will click copy, this copy button, and then moving to upload, back to Galaxy and moving to clicking on this upload button, then paste fetch data. Then we will paste data in this rectangle, and then click start. Now we have just uploaded the data to our history. As we know, we have two samples, JC1A and JB4D. So we have two samples. Each sample has R1 and R2. R1 refers to the forward read of the sample. R2 is the reverse read of the sample. We will just move to creating dataset collection. In order to create a list of dataset pairs, to just know that this R1 and this R2, the forward and reverse read belong to the same sample, which is JC1A, and this R1 and this R2, the forward and reverse read belong to the same sample, which is JB4D. We will create a list of dataset pairs. Firstly, to create this list, we will click on this check button, and then we will click on this select all, clicking then on all for selected, and then we will click on build list of dataset pairs. You can type here underscore R1, and this we can type here underscore R2, and then we will click on pair these datasets. Just we will make sure this R1 and this R2 belong to the same sample, JC1A. Click pair these datasets, and then the next one, these forward and reverse read belong to the same sample, JP4D. Then we will click pair these datasets, and then we will give it a name like input reads. Then we will click on create list of pairs. Now we are done. We have created the input reads. We can check them. The first sample has two datasets. The first one is the forward read and the second is the reverse read. The second sample has two datasets. The first one is the forward read and the second one is the reverse read. Then now we are totally fine. Now, and after getting our input reads ready, we have to run Kraken2 tool on Galaxy just to know which microbes are present in our sample or in our data. I just want you to remember what we have mentioned about Kraken2. It was our expeditions rapid scanner. It was scanning the forest for signs of life. And speaking technically, we have mentioned that Kraken2 works by breaking down DNA reads into short fragments called k-mers, and these k-mers or these fragments are then matched against a vast database of known organisms. Also, we have mentioned that Kraken2 it is super fast and maps each match to the most likely position in the taxonomy tree. These tools give us a quick, comprehensive first look at the microbial life in our sample. We have some key features here regarding Kraken2. The first point mentioned that Kraken2 it is an ultra-fast taxonomic classifier. And what does this mean? It means that Kraken2 is designed to quickly identify what organisms are present in our metagenomic sample. Like it can classify millions of DNA sequences in just minutes, making it one of the fastest tools available for this task. And about why this matters, speed is important. Speaking honestly, speed is important when we have large data sets, which is common when we work with metagenomics. So Kraken2 is highly optimized to perform taxonomic classification efficiently without compromising too much on accuracy. The second point mentions that Kraken2 uses k-mer-based DNA to DNA matching. Instead of comparing entire DNA sequences, Kraken2 breaks each read into smaller pieces called k-mers. And we have mentioned this many times. Kraken2 then uses these k-mers or these fragments and matches them against a database of known sequences. And considering why this is important because k-mer matching is much faster than full alignment methods. It's like comparing short words or phrases instead of whole sentences. It's faster, but still informative enough for our classification. The third point says that Kraken2 uses a compact reference database. This means that Kraken2 uses a compressed, space-efficient database format. It's called a compact hash table. And this makes Kraken2 too much faster to search and requires also less memory compared to traditional databases. Like you can run Kraken 2 on standard computers without needing like supercomputers. And this compact form allows your computer to handle huge databases like quickly and easily. So Kraken 2, like imagine Kraken 2, instead of saying, let me search the entire database to see if this DNA piece exists, your computer instead says, I know exactly where that piece should be. Let me check. Okay? So uh, this way is much faster. It's like there is no need to use supercomputers to handle huge databases because it is in a compact hash table format. The first point is that Kraken 2 assigns organisms by taxonomy tree. What does this mean? It means that once Kraken 2 finds where a k-mer matches in the reference database, it places that match into a taxonomy tree. If there are multiple matches, it assigns the read to the lowest common ancestor of these matches. Let me give you an example on this. Let's say a DNA read matches both E. coli and Salmonella. These are both from the same family. You already know that the family of E. coli and Salmonella is Enterobacteriaceae family. So Kraken 2 says, I can't tell if it's exactly E. coli or Salmonella. So I'll say it belongs to their shared ancestor group, Enterobacteriaceae. And this strategy avoids overconfidence. It like doesn't guess a species unless the evidence is super clear. This helps reduce false positives and gives us a reliable estimate of what's in our sample or what microbes are there. The last thing in the key features of Kraken 2 is the database that we use. It's named plus PF. This database is a pre-built Kraken 2 database. It includes bacteria, it includes viruses, fungi, protozoa, plasmids, human, and everything you can imagine. And using a wide-ranging database like this ensures you are not missing any key organisms. It just gives us a comprehensive overview of all possible microbial life in the sample. Now, after knowing like the basics about Kraken 2 tool and its key features, we will be moving into Galaxy to run our tool. So Galaxy, the first thing I will be doing is opening the training material. It will open our tutorial, and then I move to K-mer-based taxonomic assignment with Kraken 2. Now we have to follow these steps one by one. The first thing, it mentioned the tool version. We will open it. Kraken 2. This version 2.1.3 Galaxy 1. And then we will follow the steps roughly. It says single or paired reads. We will choose paired collection. Paired collection. It says here there are no compatible datasets available. That's because when I click on this input reads or on these input reads, the color is still orange. It isn't yet downloaded or uploaded. We can check if this is like a problem. We can upload our data again. Here from paste and fetch data, we will get the link to these data. Here, you will click copy and then here We will paste our links and then click start. We will wait till this turns green and then make a dataset collection or a list of dataset pairs. Or we can just make the list of dataset pairs directly. Build list of dataset pairs. New inputs. We will wait till this turns green and then proceed with our tutorial. Now our jobs turned green, like when you open the forward and reverse of both samples are now green, so they appear in the collection of paired reads here. You can see them as new inputs. We will complete to follow the step. We will complete to follow the steps through the material. Says we should just we would adjust the confidence level to 0.1. We will do this, adjusting the confidence to 0.1. It means a confidence score of 0.1 means that at least 10% of the keywords should match entries in the database. And then in a create report, we want to print a report with aggregate counts. We will click on yes. Create report. Print a report, yes. And then we will choose our database, the one we have talked many times about, plus PF. It's this one. Rebuild RefSec indexes plus PF. This version just to induce reproducibility. The same like the one in the tutorial. Let's see. Rebuild RefSec index plus PF. We will click on Run Tool. And then you have to wait until Kraken 2 runs successfully. The thing is, Kraken 2 takes some time, so you can just take a nap or you can go and make yourself a cup of coffee. I will pause till the Kraken 2 tool turns green. You see, I'll be waiting till it turns green, and then I'll be resuming our video. Hello again after our long break. Now that Kraken 2 has finished running, you could see two types of outputs. The first one is the classification file for both datasets, and the second one is the report file for both datasets. We can take a look firstly at this classification file. Maybe we can open the first sample file, JC1A. You can click on display button, and then you will see something like that. To understand every column and what it refers to, we can open this tutorial training material and click on the Gamer-based taxonomic assignment with Kraken 2, and then scroll down till reaching this. Kraken 2 will create two outputs for each dataset. First, the classification file. First, the classification file has the first column, and the first column has something like C or U. The first option for the first column is U, and the second option is C. C letter indicates that Kraken 2 successfully classified this sequence, and U means unclassified. Kraken 2 couldn't classify these sequences. Then we have the first sequence is unclassified, the second is unclassified, the third is unclassified, the fourth, fifth, and sixth all are unclassified. The seventh one is the first classified sequence in this photo. Our second column, it's just the sequence ID. It's just the ID of our sequence. Then comes the third column. The third column, which include these zeros and the 9606. This is the NCBI taxonomy ID assigned to these sequences. Since the first six sequences are unclassified, they are given 0, and then this 9606 is for the classified sequence, and it is the NCBI taxonomy ID 9606. This, by the way, refers to Homo sapiens. See that? 9606 corresponds to Homo sapiens when looking at NCBI. You can just copy the 9606 and search for it in NCBI taxonomy, and it will give you Homo sapiens. Then the fourth column is the length, it's just the length of the sequence in base pairs. 151 is the length of the first read 1, and 190, for example, is the length of the second read of the first sequence in base pairs. So 151 is the read 1 length of sequence, and 190 is the read 2 length of sequence in base pairs. After this, we have this long line. This long line. It seems kind of complicated, but it's super easy. For example, we will take the first one. This line shows how Kraken 2 tried to classify every Kamer and which ones matched specific organisms, which were ambiguous, or which are unmatched. Okay? The first one is A and colon 17. What does this mean? This means that 17 Kamers were ambiguous. Then the second number is our Kamers number, 17 Kamers, and this A letter refers to ambiguous. Okay, they were just unknown. The second one is 0 colon 15. This 15 is the number of Kamers, 15 Kamers, and 0 means that these Kamers didn't match anything. That's why we see this 0. Removing to the third one, 2055 colon 5. This means that the second number is our Kamers number, that 5 Kamers matched a species with taxonomy ID 2055. The first one means that one Kamer didn't match anything, so you can see the 0. Then this means that 5 Kamers matched this taxon ID 2220095. The last one in the first read, in read 1 of our sequence, is 0 colon 74. This means that 74 Kamers didn't match anything. And about this is just a separator. This is just a separator. I can't select it. This is just our separator. Okay? That separate read 1 from read 2. Then we move to the second read 2. This, the first one is 0 colon 3. This means that 3 gamers didn't match anything. Then after this, A colon 53 means that 53 gamers were ambiguous. Then 2 to 2 means that 2 gamers matched taxon ID 2, and so on. And for this example, that's written here, the same as we have said, the same way. 562 colon 13, it means that the second number is the number of gamers. It means that the first 13 gamers mapped to taxonomy ID 562. Then comes 56124, means that 4 gamers match taxon ID 561. Then after that, A231 means that 31 gamers was or contained an ambiguous nucleotide. A is for ambiguous. Then the last, then comes 0 colon 1, that one gamer didn't match anything. The next gamer wasn't in the database, it didn't match anything, so you can see the 0. After that, 562 colon 3, this means that the last 3 gamers mapped to taxonomy ID 562. And as we said, this indicates that that is the end of the first read and the start of the second read for bird reads. They gave us here like three questions. The first one says, is the first sequence in the file classified or unclassified? Here, it's unclassified. We have said that the first 6 sequences are unclassified. The 7th one is classified, so you know the first one is unclassified. Then the second question is, what is the taxonomy ID assigned to the first classified sequence? This is the first classified sequence, the 7th one. What is the taxonomy ID assigned to the first classified sequence? You can see the taxonomy ID here in this column. In the third column, it is 9606. So that is the answer for the second question. And then comes the third question. It asks about the corresponding taxon. The taxon ID is 9606. When you insert this in NCBI, you get Homo sapiens. And that's the answer for the 3 questions. You can see this, the classification file for the full sample, JC1A, here. You can like scroll down till the end. This report file is so simple. You can find it here. This, we can check, for example, the first sample report file as an output from Kraken. This will be what we see. We can check tutorial to like understand what the first column means, the second, the third, and so on. I just want you to focus on the first column, which is this. The first column, this one, with these percentages. It is the percentage of total, like how common this group is found. For example, the bacteria is found by this percent, 12.58. So 12.58 of our entire sequences is or does belong to bacteria. Then the second column, this one, the second column, we can like see it clearly from here. The second column, this one, column 2, it's just the total reads, the count for this group. For example, the count for bacteria and all its subgroups is 17256. This is the total reads, which are classified as bacteria. Then the third column is the direct reads. Then comes the fourth column, which is so important. I want you to focus on the first column and the fourth column. The fourth column, for you, U refers to unclassified. R refers to root. D, for example, refers to domain. B, for example, refers for phylum. G for genus, and so on. It is the rank code. Then we can get back to our tutorial. It will ask us some questions to see whether we understand this or not. Just before going through these questions and trying to answer them, I want you to know, or I just want to ask you, have you ever asked yourself what is the difference between these rank codes? Why Kraken 2 has assigned some sequences to domain level and other to class level or order or genus? What is the difference here? What makes this difference? As we have mentioned before when explaining in details what is Kraken 2 and what is its key features, what are its key features, we have mentioned that Kraken 2 avoids overconfidence. It doesn't guess species unless the evidence is clear. And if there are multiple matches, it assigns the read to the lowest common ancestor. This means I can give you an example so that you can understand this simply. Imagine you are at a big family reunion and you have an old relative you haven't seen for a long time. This relative spots you and says you're definitely from the Omara family. That's my family name. Okay, based on some facial features you have. So, one of my old relatives that I haven't seen for a long time spots me and said, you have facial features like the rest of your family. So, this relative is 100% sure that I belong to family Omara. Okay? But he isn't sure about me belonging to a certain subfamily. Like he isn't sure that I belong to Adham's subfamily. Adham is my father's name. So this relative is sure I belong to Omara, but he is not sure that I belong to Adham. This is the same Kraken does. Okay? So Kraken 2 is sure these sequences belong to bacteria, but isn't sure these sequences, for example, belong to Enterobacteriaceae. And that's it. Moving towards these questions, the first question, it just said what are the percentage on unclassified for GC1A and GB4D. This is for GC1A, JC1A. 76.85% is unclassified. This should be 76.85. Approximately 77% is unclassified for JC1A, okay? Moving to JB4D file, we can just click on this, report file for JB4D. 89.81% is unclassified. Then that is the answer. 89.8 is unclassified. Approximately 90% is unclassified for JB4D. The second question asks about the kingdoms found for both samples. For example, you can find here bacteria with 12.58%, approximately 13%. And then you can find eukaryotes with 9% and viruses with 0.03%. You can check them here from display button. You can check the report file on JC1A. It's uploading. You can find here the bacteria 12.58. Then we can search for eukaryotes. This eukaryota, it's 9.39%, and then viruses. Viruses is found in 0.03%. What about JB4D? Let's display it. 89.8 is unclassified. Bacteria is found with 9.22%. We can search for viruses. For example, there are no viruses here. For eukaryotes, they are not here also. So we can move back to our tutorial. It says 9% bacteria and 0.7 eukaryote. We can search for it again. This here, 0.65% eukaryote. Okay. Back to the tutorial. It asks about where might the eukaryotic DNA come from? It's likely a human contamination, and this is very common when it comes to lab methods or lab treated samples. The first question asks about the diversity of proteobacteria in JC1A and JP4D. Regarding proteobacteria, it's found in 8.77%. You can simply answer this question based on the report files. Let's click on display. On the report file of JC1A, you see here proteobacteria, and then you see class alpha proteobacteria, and we can search for word proteobacteria to find different classes which belong to proteobacteria. We have here alpha proteobacteria with 4.94%. We have then beta proteobacteria with 1%. We have gamma proteobacteria with 0.6%, and then delta with 0.15%. So there is some kind of diversity here in proteobacteria classes. Moving to JB4D, we can see this is proteobacteria with 8%, and this is alpha proteobacteria with 6%. We can search for it, for proteobacteria. We can see then beta proteobacteria, it's just too low, 0.13%, and we have gamma with 0.05%, and then delta with 0%. So you see there is more diversity in JC1A file, report file, okay? There is here more diversity in JC1A report file. You can find 5% of alpha proteobacteria. You can find beta, I guess, with 1%, and you find gamma and delta with reasonable percents or reasonable percentages. And then let's move back to our answers or our solutions. It says JC1A seems to have a big diversity of classes, and that's what we have proved. Now we are done with Kraken 2. Moving towards Kraken, we will not explain everything in details. We will just explain 2 points. let's move to our slides to explain them and then be back to Galaxy to apply them. This is Bracket Tool, our abundance estimator. I've said that we will mention like 2 points only, but here are 5 points that we need to discuss. But I promise they will take 4 minutes maximum. The first key feature of Bracken tool is that it refines Kraken 2 classification output. Bracken 2 doesn't do classification on its own, like it doesn't do classification from scratch. Instead, it takes the results Kraken 2 gave you, the list of microbes it found, and improves them. So Kraken 2 improves the output of Kraken 2. Especially when Kraken 2 wasn't confident at lower levels like species. For example, if Kraken 2 is our old relative that we like haven't seen for long years or for long time, Bracken here represents your uncle that you are seeing on a daily basis. Okay, so he knows you so much, and he is confident that you belong to the subfamily, or he is confident you belong to a certain type of species. Okay? So, if Kraken 2 is your old relative, Bracken 2 is your uncle that you see on a daily basis, so he is more confident that you belong to your fathers or a subfamily. The second point in Bracken is that it uses Bayesian statistics to re-estimate species abundance. This just means Bracken uses a smart mathematical approach, which is Bayesian probability, to better guess how many of each microbe are actually there, even if some reads are unclear. It uses probability to estimate or to put an estimate of how many microbes are there. The third point is that Bracken redistributes reads more accurately across taxonomy. Sometimes Kraken 2 assigns a DNA read to a higher level, like genus, because it's not sure about the species. This is where we can give our example, the old relative and your uncle that you see on a daily basis. Here comes it. Bracken tries to reassign those reads more specifically down to the right species, given a more precise count. Okay, so it redistributes reads more accurately. Your uncle knows you and can like match you to your father. He is confident and he will match you to your father in the right way. The first point is that Bracken improves species level resolution. As a result of this redistribution, Bracken gives you a much better picture at the species level. That's what I want you to know. The fifth point and our last point is that Bracken requires same database as Kraken 2. You will see when we are going to apply Kraken 2, when we are going to apply Kraken 2 in Galaxy, we will be using the same database we have used for Kraken 2. Let's move to Galaxy and apply what is said or what's mentioned in the tutorial to run Kraken tool. We will be opening our material or our tutorial. This part, estimate species abundance with Bracken. We will click on this tool. This is Bracken tool to re-estimate abundance at taxonomic level from Kraken output. The thing is that we don't use single datasets. We use dataset collection. So we will click here and we will choose our Kraken 2 report file, Kraken 2 output report file 28. We will choose it and then we will follow the other adjustments or the other steps mentioned by the tutorial. It said Kraken report file, report output of Kraken, and this is what we have done. Then we will select a K-mer distribution. Our database, we will use the same database we have used for Kraken. It is the pre-built DIRSEC indexes plus PF and this version. We will use the same database used for Kraken 2. It's like using a different map to navigate the same forest. Things won't line up. So we have to use the same database we have used for Kraken 2. We will adjust level to species as it is, and then we will click on this, produce Kraken style Kraken report. Yes, we will change it to yes. We will make sure everything the same as here. Level species, produce Kraken style Kraken report, yes. So that's it. We will just run the tool. And we will be waiting for Kraken tool to just produce the output. It will take some time, but less time than Kraken 2 file or Kraken 2 output to be produced. So we will wait till this turns green, and then we will see its output and compare it to Kraken tool or Kraken 2. It just took 1 minute to become green. So here our two outputs from Kraken tool. I want you to focus on this Kraken on collection report file, and we will open this report file. We will display it, for example, the report file of Kraken for JC1A sample. You will see here the name, and then followed by taxonomy ID, and then taxonomy level, and then Kraken assigned reads, like the reads Kraken has successfully assigned, and then the reads added by Kraken. For example, let's look at Bradyrhizobium species SK17. In these species, as example, Kraken 2 has successfully assigned only 40 reads to this species. Like, give this an attention. Kraken has assigned only 40 reads to this species, Bradyrhizobium species SK17. Not because it's rare, but because Kraken 2 gets stuck at higher taxonomic level when DNA is ambiguous. Then, this is why we use Bracken tool. Bracken tool fixed this. It analyzed those uncertain reads and determined 1382 reads, and it assumed that they actually belong to this species, Bradyrhizobium species SK17. That is 35 times, that's approximately 35 times more than Kraken 2 detected. And this is why we use Bracken. We use Bracken because it rescues or saves our reads, saves the reads Kraken 2 leaves behind. It turns the unclear genus level data into precise species count. Thus, it reveals the true abundance of microbes in our sample. We can move back to our slides. Just see this. Take a look and focus. Kraken 2 has assigned only 40 reads to this species, Bradyrhizobium species SK17. Kraken has assigned 1422 reads to this species, which is 35 times more than Kraken 2 detected. Look at this. This is just a selection from the report output of Bracken tool. You can see Bradyrhizobium species SK17, thesis, 40 reads, Kraken 2 called for SK17. Turns out, Bracken found more, it found 1382 more reads hiding in plain sight. It's like Kraken 2 gives us a blurry photo and Bracken sharpened it into HD. That's it. Now that we have found some microbial life in the forest, using our tools Kraken2 and Braken for identification of microbes and then estimating their abundance. We need a map now to understand where everything fits. That's where Corona comes in. Corona builds a magical circular map that lets us zoom in the microbial tree of life from kingdom all the way down to species. The same as this, from kingdom all the way down to species. Moreover, we use it to visualize our Kraken data after a quick conversion and metafilan results, helping us see which microbes dominate and where they fit in the bigger biological picture. Now, with the key features of Corona, our magical map, the first one is that Corona creates an interactive, zoomable circular chart, the same as we have seen. We will see it after getting after applying Corona on Galaxy and getting our outputs ready. Corona turns here our microbiome data into a circular chart where you can zoom in from broad categories like bacteria to specific species. This is so important because it helps us visualize and explore our results. Also, this magical map can be shared. The next point is that Corona visualizes taxonomy from kingdom the way down to species. It just shows taxonomic hierarchy clearly. That means you can visually trace the microbial family tree from domain to phylum to class and so on. And this is important as this is an evidence that Corona makes complex data easier to understand. And instead of just having some columns with numbers and identifications or taxonomy ID, we will have a circular map where microbes can fit to imagine where they lie in the greater picture, in the greater biological picture. Then the third point mentions that Corona works with metafilan or Kraken output. And you have to focus on this. Corona works with Kraken output, but after a slight conversion. And we will see this when applying or running the Corona tool on Galaxy. And it works with metafilan output directly. The first point is that Corona is easy to explore, makes it easy to explore diversity and structure of the community. Um, this doesn't need any clarification. It's just clear. So this is our Corona. We can move to Galaxy to run the tool. We will just open our training material and proceeding to this visualization of taxonomic assignment. It just mentions that there are several tools for visualization of taxonomic assignment like Corona, Finch, and Bavian. We will focus on Corona and Bavian for this tutorial. The first thing, it says that Kraken outputs cannot be given directly to Corona, so they need just a slight modification. We need to convert Kraken output using this tool just to make it suitable to be given to Corona for visualization. We will click on this tool, just as simple as that. And then we will choose dataset collection instead of single dataset. And then we will choose the Kraken2 report file, this. And then click run, as simple as that. You will wait for this to turn green, for Kraken2's file output to turn green. It just took 1 minute to run successfully. So we will visualize this output. For example, we will go for the first sample, JC1A. and click on display button, you will have something like this. And what does this mean? You just have like two columns. The first one is the number of reads, or how many reads or fragments were found for that group. For example, 105397 reads or fragments are unclassified. Then we have 3869 reads where belong to kingdom bacteria, and then we have 2868 reads belong to phylum Proteobacteria, and so on. So the first column is how many reads or fragments were found for that group, and the rest columns, column 2, 3, 4, 5, and 6, and the rest are just they just show the taxonomy. They just show the taxonomy ranging from kingdom, passing by phylum, class, order, family, genus, and then species at the end. We will go to our training material. This is the output that we have just seen. It asks us two questions. The first one asks about the different columns. We have already mentioned that the first column is how many reads were found for each group, and the rest of the columns are just showing the taxonomy level. What are the lines? The line is just a taxon with its hierarchy and the number of reads assigned to it. So, for example, if we take this line to discuss it, we have 10 reads assigned to the species Paracoccus pantotrophus. If we took this other line as an example, it tells us we have 53 reads assigned to genus Paracoccus, and so on. We will proceed to Corona visualization. We will just open this tool, Corona pie chart tool, and then it gives us like two steps to be done. The first one is to change type of input data to tabular, and the second one is the input file we will insert. It is the output of Kraken tools, this. So we will change the type of our input data to tabular, and then we will choose dataset collection, that is the type of our input file, and this. Then we select the convert Kraken tools convert Kraken report file. The file we have just produced an output for, and then we will click run tool. We will wait for this job to be done, to successfully run, and then we will proceed with discussion of the chart that's produced. The job turned green in just a minute or two, so let's proceed and visualize our Corona output. It just gives us this circular chart. It's just a visualization of Kraken 2, so it is the same percentages. Like 78% of the first sample sequences is unclassified. Then we have this percent of bacteria, we have this of eukaryota, and then we have 0.03% belong to kingdom viruses. This is for the first sample. You can zoom in and out from categories like bacteria, all the way down to species. You can zoom like this, just clicking 2 clicks on kingdom bacteria. Then you have Proteobacteria here. We can zoom in. You will find that Proteobacteria, as we have previously mentioned, is divided into Alpha Proteobacteria class. Then we have Beta Proteobacteria class, and then Gamma Proteobacteria, and then Delta Proteobacteria. These are the 4 classes. forming the phylum Proteobacteria. We can zoom an alpha Proteobacteria class, for example. You will see that alpha Proteobacteria has a family like Bradyrhizobiceae. We can zoom in, and then you will find Bradyrhizobium genus. We will zoom in, and then you will find the species we have already talked about when mentioning the difference between, or when doing a comparison between the output files of both Kraken 2 and Bracken. This is species Bradyrhizomia species SK17. It constitutes the bigger part of Bradyrhizobium genus, more than the other species, the other two species. And that's it. Shifting towards the other sample, we can visualize it as simple as that. We will zoom out from here, and then the phylum, then to the root directly. You will see that 90% of this is unclassified. This is the second sample, JB4D, corona output, or corona chart, circular chart. You will find this bacteria. We can zoom in like that. You can find alpha Proteobacteria also, beta Proteobacteria is found, and so on. You can zoom an alpha Proteobacteria, and that's it. As simple as that. Now we are done with the corona pie chart. We will just proceed to the training material to see if there are any questions on this part. It just asks the same questions we have already seen, or we have already answered when we were talking about Kraken 2 output, Kraken 2 report file. There are the same questions as corona is just a visualization of corona corona is just a visualization of Kraken 2 output, but after a slight conversion. After conversion of Kraken 2 report file using Kraken tools, this tool. We will proceed to discussing the second visualization tool, Pavian, from our slides. Just very simply, Pavian is our detective's board, where we pin up clues from both samples and compare them side by side. Pavian pulls in data from Kraken tool and Metagenomic and gives us interactive tools like Sankey diagrams, like bar charts, like comparison tables, and so on, to help us see differences between our samples, the undisturbed one and the one we have intervened in. These are the key features for Pavian tool. The first thing, it said that Pavian is an interactive tool to compare samples. It just lets us look at multiple samples side by side, showing what microbes are found in each of them. And this is important as it helps us compare the treated sample versus the control one, or the healthy versus diseased, very clearly or in a very clear manner. Pavian is great for exploring results from Kraken and Metagenomic. Just use reports from Kraken and Metagenomic, and that's it. The third point is about the interactive tools Pavian produces. It means that Pavian shows result using some visual tools, like Sankey diagrams, like bar charts, and others. The Sankey diagrams, it just flows from high-level taxa like domain to specific species with showing proportions. And then the bar charts just compares how many reads matched each group across different samples. And this makes it easy to see which microbes increased, which decreased, and which stayed the same and isn't affected by our intervention. The fourth point is based on the third. It just tells us that Pavian helps us see the differences between the control and treated sample, and that's what we have already mentioned. And the last point, it says that Bayesian is ideal for clinical or ecological interpretations. Bayesian is just a powerful tool for asking deeper ecological or health related questions, as it helps us like make a comparison between the control and the treated samples. It helps us compare between the healthy individuals and the diseased one. And that's it all about Pavian. We will just move to Galaxy to run the tool. We will just move to Galaxy training material and then scroll down to visualization using Pavian. It says that Pavian is an interactive visualization tool for metagenomic data. The thing is that we will move to the step of running Pavian. We will just click on the Pavian icon, this tool, and we will select or click dataset collection. We will choose the Kraken 2 report, Kraken 2 in collection report, and we will click run tool. As simple as that. We will wait for Pavian to turn orange. Look, Pavian, it tells us that Pavian is an interactive tool, runs a Galaxy interactive tool, and we can access it when it becomes orange. So we will wait for this job to turn orange, and then we can see our results. The job turned orange in just a few seconds. You will click on display. It will direct us to another page. First, we have to import data through these steps. Step number 1, we will click on use data on server. You'll click on use data on server. Step number 2, we will select both samples and click on read selected directories. You will select both samples and click read selected directories. We will ensure our table looks like this. Check you have a table in available sample sets looks like this. Yes, we have this table. And then we will click on Save Table. Then we will move to Results Overview. And this is the output here. This is the same output that we have generated. Through these questions, you will understand this table. The thing is that you can see the same numbers in Kraken report file. Because the same thing as Corona, it's just a visualization of Kraken report file. Okay? You have here the number of reads for both samples, and they are different from each other. Sample JP4D has higher reads or more reads than the other sample, JC1A. And here you see the classified reads of both samples, the percentages of classified reads. And here you see the unclassified reads, the same percentages we have in Corona and in Kraken report file and so on. You have unclassified reads 90% in the second sample, JB4D, and 76.9, approximately 77% in the first sample, JC1A. And you have microbial reads or bacteria with 13% in the first sample and 9.4% in the second sample. And then you have this is microbial reads, sorry, and this is bacterial reads. And here you have viral reads. It was 0.03% in the first sample, and it was 0.0004% in the second sample. And this is also what Corona gives us. It is the same output as Corona, the same numbers. Then we are going to answer these questions. Does both sample have same size? No. The first one is uh, bigger than the second one. It has more reads. The answer said JB4D has much more reads than JC1A. And the second question asks about the percentages of classified reads for both. This is the percentages of classified reads, 23.1 for JC1A and 10.2 for JB4D. This is the same percentages we have here in our output. And then it asks about the percentage of bacterial reads. Are they similar? No, they are not similar. You have here bacterial reads with 12.6% in the first sample and 9.22% in the second sample. And this is what's written here. We can move now to inspecting samples separately, like each sample separately, using Sankey plot and visualize it using Sankey plot. From here, we will click on sample. This will be shown. This is the Sankey plot or Sankey diagram. It flows from high-level taxa, like domain, to specific species, like, for example, Rhizophium species SK17 that we have talked about many times. When we click on it, you see it is like a comparison between Rhizophium species SK17 across the two samples or between the two samples. The first sample, JC1A, shows higher reads than JP4D. You see here are 40 reads and here is only one read. Here is the domain and here the kingdom, phylum, family, genus, and finally species. We can compare proteobacteria across both samples simply by clicking on this proteobacteria. It will show us the different proportions of proteobacteria across the two samples, JC1A and JP4D. It's so much higher, just more reads in the second sample, JB4D, much higher than the first sample, JC1A. And that's it. We can compare the proportions of any of these between the two samples, side by side. Back to our galaxy. It just asks about what is Sankey diagram. Sankey diagram is a visualization used to depict a flow from one set of values to the other. What are different set of values represented at the horizontal axis? This is the horizontal line. It just started with domain, followed by kingdom, then phylum, then family, then genus, and ends by species. Domain on the left to the species on the right. That's what we have already done. We clicked on proteobacteria in the Sankey plot and then inspected the graph on right and compared both the proportions in both samples. It asks about the number of reads assigned to proteobacteria, similar for both samples. Are they similar? No, they aren't. JB4D has many more reads assigned to proteobacteria than the first sample, JC1A. Here, JB4D has many more reads, higher reads than the first sample, JC1A. The second question is why? Just because like a larger number of reads originally in JB4D originally matched to proteobacteria. It's simple. It's like counting how many puzzle pieces match a certain color. If JB4D has more pieces that fit the proteobacteria color, then It simply has like more of them in the sample. It's logical. We can now move to an inspection of samples with Bayesian to this part. Through this, we can compare both samples in terms of different levels like domain, like class, like phylum, like kingdom, and so on. Just by following these steps. The first step tells us to click on this comparison button on the left panel, and then to compare the percentages instead of reads numbers or like row reads. We will click on this percentage icon or percentage blue button, and then we will unselect or unclick reads, and then to compare both samples in terms of domains, in terms of different domains, we will click on domain. So you see, bacteria is much higher in the second sample, and eukaryote is much higher in the first sample. These are the most important findings here. So keep in mind, the first sample has higher eukaryote, and the second sample has higher bacteria, much higher bacteria than the first one, was 93.41%. The viruses is percentage is lower in both samples, and the RK is low in the second sample, and it's not found in the first sample. Okay, so this is the comparison between both samples in terms of different domains. Okay, then it asks us about is there a similar proportion of bacteria in both samples? No. JB4D sample has much higher bacteria than the other sample with 93.4%. Look, after removing the Homo sapiens, First, I need you to remember that the first sample is the sample from the unfertilized bond, the bond we have never intervened in, we have never made intervention, the control bond, and the other sample is from the bond that we have added nutrients to it, we have fertilized this bond, okay? So, the bond with fertilizers or with nutrients, with added nutrients, has much higher percentage of bacteria. Before removing the Homo sapiens, before removing or filtering out the Homo sapiens. Look, after filtering out Homo sapiens, we will filter taxa, here, Homo sapiens. You will see difference. Just waiting for the internet connection to come back. Fortunately, the internet connection came back. So here we can see the difference after removing the Homo sapiens, after removing like the human contamination from the samples. You see that JC1A have now higher percentage of bacteria, which is approximately similar to the second sample, JP4D. And regarding why, this is because after removing the human contamination, the first sample was highly contaminated with human. It has high contamination from human sources. So after removing the human contamination, after filtering out the Homo sapiens taxa, it's likely it become higher in bacteria percentage, just like the other sample. This means it just had high contamination level from human sources. It says we can select Homo sapiens as a filter taxa to remove the Homo sapiens or the human contamination from the samples. And it asks us a question, is there similar proportion of bacteria in both samples after removing the Homo sapiens? It says after human filtering, both samples have similar proportion of bacteria, and this is what we have already proved. Then we can click on class green button to see the differences, but regarding class, in terms of class levels, you will see the second sample, JB4D, has higher percentage of alpha proteobacteria, and the second sample, the first sample has somehow high percentage of alpha proteobacteria. It has also 13 percentage of beta proteobacteria and 7 of gamma proteobacteria, 2 of delta proteobacteria. So you see there is less diversity in the second sample. It's just full, or it's just very high in alpha proteobacteria and low in the others. There is no diversity in the class level or on the class level. But the first sample, JC1A, it's slightly distributed all over some classes like alpha proteobacteria, beta, gamma, and delta. It asks us about the diversities of classes in both samples. As we have already said, JB4D seems highly dominated by alpha proteobacteria class. There is no distribution. It's just concentrated in alpha proteobacteria. And the first sample, JC1A, has a majority of alpha proteobacteria, but also significant proportions of the other classes like beta proteobacteria, gamma, and delta. The second question is, what could it biologically mean given that JC1A is the control and JB4D is a sample from the fertilized bond? We have already mentioned that. It just says that alpha proteobacteria seems to have survival advantage in the new environment. JB4D is from the fertilized bond and it's high in alpha proteobacteria. This means that alpha proteobacteria has survived these conditions, has survived the fertilizers, high nutrient availability. So that's it. Now we have finished deviant tool and we will go through metaphylan and then we will tell what's the most important points to mention in this tutorial, take home messages, and that's it. Let's go to our slides to discuss what's metaphylan and how we can use it. Metaphylan, as we have previously mentioned, it was like our expedition magnifying glass. It doesn't try to scan everything, instead it zooms in on very specific DNA clues that are unique to certain microbes. Using millions of carefully chosen marker genes from over 27,000 species, it gives us extremely accurate results. It acts like identifying an animal just from its unique footprint. Unfortunately, as we have previously said, metaphylan is way slower than Kraken 2 and it might miss microbes that aren't in its database, but when it says something is there, you should be trusted. Regarding these key features, the first one mentioned that metaphylan profiles microbial communities from metagenomic shotgun data. This means that it tells you what microbes are in your sample using the DNA from all the organism, not just bacteria like 16S sequencing. The second point tells us that metaphylan uses clade-specific marker genes. And this means that instead of looking at all DNA, metaphylan only looks for unique markers that are known to belong to specific microbes. And this is so important because this makes the results very accurate. Like I have said, when metaphylan says something is there, you should trust it. The third point is that metaphylan estimates relative abundances. It doesn't just tell you what is there, it tells you how much of each microbe is in your sample. The last point, or the point before the last one, it says that metaphylan covers bacteria, archaea, viruses, and eukaryotes. It just offers a species level and sometimes even a strain level resolution and covers all these domains like bacteria, archaea, viruses, and eukaryotes. The last point mentioned that metaplan is of high accuracy, but the negative point is that may miss rare or unsequenced data or unsequenced organisms. Metaplan is great at identifying microbes with known markers. But what if the species is rare? What if the species aren't found in the reference database? Metaplan unfortunately won't be able to detect it. And this is like important in some form. It's like important because this makes Metaplan trustworthy. When it says something is there, it's actually there. Let's move to Galaxy to show how the tool works. We just have to scroll down or just click on this marker gene based approach using Metaflan. And we have to know that Metaflan in Galaxy cannot directly take the paired collection as input as its input file, but expect two collections, one with the forward data and the other with the reverse data. So before launching Metaflan, we need to split our input paired collection. The first step, to split our input paired collection, we will use this tool on the collection. We will click on it and then choose our input data and then click Run Tool. We have our output ready. Both are ready. So we will go back to our training material. The next step is Metaflan tool, is running Metaflan tool with adjusting these parameters. We will click on this tool, Metaflan, this version, and this, and these parameters. We have to change this to paired end files. And then we will change this single dataset to dataset collection and choose our forward one. This forward paired end FASTA or FASTA-Q file with microbiota reads. This is the output of our unzip collection or unzip reads. And then we have the reverse. We will follow the same steps. We will choose dataset collection and then reverse. And then in outputs, output for Corona, we have to adjust this to yes. This is so long. Output for Corona, we will click yes. And then we have to run the tool. We will wait for Metaflan output to be produced. And then we are going to discuss it. Metaflan outputs are finally ready, so we can check them one by one. The first one, and the most important one, is this output, predicted taxon relative abundances. It's just a table showing which microbes or taxa were found and their abundance. We can display it. For JC1A, it's null. It's empty. You can see this. All of the sample is unclassified. 100% of the sample is unclassified. So we can check the other sample, JB4D, can display. And then you see we have first colon, and then we have second colon, third colon, and this fourth colon named additional species. Our first colon just shows the full name from kingdom all the way down to species. And then our second colon, as it's written here, it's the NCBI tax ID. There's the NCBI taxon ID number for each taxon. on NCBI. Then we have the third column, the relative abundance, the percentage of reads matching that taxon. And then we have the fourth column named additional species, and it's sometimes blank. That's it about the predicted taxon relative abundance. So for example, bacteria kingdom relative abundance is 100%. So the sample as a whole belongs to kingdom bacteria. 100% of our sample belongs to kingdom bacteria. And then another example you have here, Proteobacteria phylum. 5.6% of your sample belongs to phylum Proteobacteria. Then we have this as another example, class Alpha Proteobacteria. The same percentage as Proteobacteria, 5.6% of our sample belongs to Alpha Proteobacteria, and so on. So this is all about the first file produced by Metafilan named Predicted Taxon Relative Abundances. This is the main result, tells you what's in your sample and how much of it. Then moving to the second output, we will go for this predicted taxon relative abundances for Corona. It is the same information, it contains the same information as the other output we have discussed, predicted taxon relative abundances, but it's more suitable, it's a format ready for Corona visualization. You see here, it's written for Corona, so it is a format ready for Corona visualization. We can display it. You will see the first sample, 0 lines, 0 colons, because all of this sample is just unclassified, so there is nothing to talk about. And moving to the second sample, JB4D, we will display, click display button. You will see something like this. The first column tells us percentage of read for the taxon. And then from column 2 till the last column, column 9, you will have like taxonomic levels from kingdom all the way down to species. That's it about this file. It's just the same as the first file we have discussed, predicted taxon relative abundances, but it's a format ready for Corona visualization. Moving to the next output, we can see here biome file. This biome file contains the same data as the table, but in biome format. And this format is used in many other tools like Mother, like Chime, and so on. It's very common in microbiome studies as general. And then we have this file. We will go for this file. It is SAM file. SAM file means sequence alignment map file. It just shows how our reads were mapped to the Metafilan database. And it's usually used for advanced analysis, not the basic analysis we are going through. So that's it about SAM file. And then we have this output, POTI output. POTI, it's just SAM file with mapping and this is used mainly for like advanced analysis. We've just mentioned all of this information, so we will go for these questions. The first questions ask us about the kingdoms have been identified for the first sample, JC1A. And honestly, there are no kingdoms because all of the reads are unclassified. All reads have been unclassified, so no kingdom identified. The second question, it asks about the kingdoms identified for JB4D, bacteria. We have already seen this. We can check it from our most important file. This bacteria. So yeah, it's bacteria. Then the third question, how is the diversity for the second sample JB4D compared to Kraken results? You can see that diversity is really reduced using metaphylan compared to Kraken. As we have already mentioned many times, metaphylan is 100% accurate. When it says something, it's really there. But Kraken has some form of inaccuracy. So when it comes to diversity is more in Kraken compared to metaphylan. And maybe this is due to metaphylan restricted database. We will move directly to generation of Corona visualization for metaphylan output. So we will go to this tool, Corona pie chart, and then we will follow the steps or the instructions. Type of input data, tabular. Input data, we will select tabular, and then our input file will be, as you see, the Corona output of metaphylan. We will select dataset collection, and then we will find the Corona output for metaphylan, this one. And then we will collect on run tool. We will wait for the results till this job runs green. We'll be waiting. You can see our job ran successfully. So we are going to display it, clicking on display button. You now see a blank page when we click on the first sample. This first sample shows a blank page. Why? Because the sample as a whole is unclassified. There are no classified reads in this sample, the first sample, JC1A. We will move and click on the second sample, JB4D. You will see something like this. You can see less diversity than Kraken tool output. Let's go back to Kraken tool output so we can compare. You can see the difference. This is the second sample. It's much more, there are much more diversities here, variations. You can see here this species, Phycocomus zingai. It represented, I guess, 5.6%. We can see this from this file, relative abundances. Phycocomus, this, the last line. Both of them represents 5.6% of the whole sample reads. We can go back to our chart. These SGB followed by number, they are like species. These species are not officially named, like they don't have an official scientific name till now. So that's it. We can move back to our training material. It just mentions that, as pointed before, the community looks a lot less diverse than with Kraken, and this is what we have already mentioned. This is probably due to the reference database, which is potentially not complete enough yet to identify all taxons. This database is regarding database of metaphylan. It's not complete enough to identify all the taxons. The second reason may be that there are too few reads in the input data to cover enough marker genes. So, no taxons has been identified for JC1A, and there are much less reads than JB4D for JC1A. Okay? However, Kraken is also known to have high number of false positives. You know, Kraken is not that accurate, but Metaphylan is super accurate. So each one of them has its pros and cons. And that's it. We can just move to choosing the right tool part. And then we will be finishing our video for today. Regarding choosing the right tool, the thing is that when it comes to taxonomic assignment, while analyzing metagenomic data, Kraken 2 isn't the only tool available. So, several papers do benchmarking of different tools. If you don't know what this benchmarking thing is, benchmarking in general just means testing and comparing different things to see which one of them performs best or performs better. Okay? When applying this on metagenomics, benchmarking tools means scientists are checking which tools are best at identifying microbes in a sample by testing all these tools on the same data and then comparing the results to see which is more accurate, which is more fast, and so on. We can move to our slides just to simplify this benchmarking thing about why we compare tools, just to see which tool best identifies microbes in metagenomic sample, like which tool is faster, which tool is more accurate, and so on. Regarding the second point, how are tools tested, like how this benchmarking thing works, we have two ways. The first is the CAMI challenge, and CAMI refers to the critical assessment of metagenome interpretation. And this just means that many labs got the same test dataset, and then each lab ran their tool on it and sent the results back, and then the results were compared. Like what's written, many labs analyze the same data or the same sample and share results. The second is Ye and his research team in a study published in 2019. This one research team did everything themselves. They ran all the tools on known datasets, and then they compared the results. What is in the test data? For the CMI challenge, they have used the CAMI datasets. CAMI datasets are more difficult, as like only 30 to 40% of the read are from known microbes. The rest are just unknown or are from new microbes. They belong to new microbes. So it's kind of a harder test. The second dataset is IMMSA, which he and his research team used. It's easier as microbes already are well described. Microbes are datasets, all microbes in these datasets are already known. So tools usually perform better here. We can move to the next slide to just see how do they check which tool is better, like based on what, how performance is measured. Performance is measured based on 4 points. You can think of it like grading each tool with different questions. The first one is regarding the precision. We can just ask, of all the microbes the tool said were in the sample, how many were actually correct? How many identified microbes are actually correct? The second one is the recall. We can ask like, of all the microbes that really were there, how many did the tool successfully find? Like how many real microbes were found by the tool? The third one is the L2 distance. Did the tool get the right amount of each microbe? Not just names, but also how many of each. How close the tool's microbe counts are to the true amounts. And the first one and the last one is the precision recall curve. This just shows the balance between catching as many as real microbes as possible, catching as many real microbes as possible, that is the recall, and not getting too many wrong ones, and this is the precision. It just balances precision plus recall to show overall performance. And that's it regarding the choosing the right tool part. We can just summarize this part, choosing the right tool, before moving to our tutorial, to just finalize this video. The thing is that scientists wanted to know which tools are best at identifying bacteria, so they tested many tools using sample data. Some are easy and others are hard. They checked how many correct microbes each tool found, how many it missed, and like if it got the amounts right. And that's how they figured out which tool works best. That's it regarding choosing the right tool part. We can move to our tutorial just to finalize this video. Now we have discussed this, but just in simpler terms, in simpler words. So we can move to this table. Let's just take a quick look at some popular taxonomic classification tools and what they are best for. Here we have the first tool is MOTUS. It's not available in Galaxy, but it's one of the most memory efficient tools out there. Then you have Metafilan. We have already talked about Metafilan in details. It uses marker genes and it's surely available in Galaxy, and it's recommended. It's a great choice if your computer has low memory. It can work well with less than 2 gigabytes. Then the third tool is Dudes. It isn't available in Galaxy, and then you have Focus. It isn't also available in Galaxy, but it's fast and memory efficient. It's memory efficient. Okay. Then we have Bracken and Kraken, and we have already mentioned Bracken and Kraken in details. Both use gamers, and both are fast. But talking about Kraken, Kraken is the fastest of all. It performs really well with big data sets, but unfortunately, it needs a lot of memory, like over 100 gigabyte when using custom databases. So, depending on your needs, like speed, like memory, or available tools, you can choose the ones that fits your data best. And that's it about choosing the right tool for your data. Here we are going to finalize our video. Let's go back to our slides, to the last slide. In this tutorial, we explored how to perform taxonomic profiling of metagenomic data using Galaxy. The key idea to remember is taxonomic profiling helps us understand who is there in the microbial community, who is there in a metagenomic sample. We have explored tools like Kraken2, Bracken, Metafilan, Pavian, and Krona. We applied Kraken2 and Metafilan to assign taxonomic labels to our microbiome sample datasets, and we then have visualized our results using Krona and Pavian to just analyze and compare those datasets. Finally, we have discussed the important facts when it comes to choosing the right tool for taxonomic assignment. And then you already know now that using Kraken2, Bracken, Metafilan, Pavian, and Krona, these tools work together to classify metagenomic reads, estimate microbial abundance, and visualize the microbial composition. Whether you are exploring a forest soil sample or gut microbiome data, these tools help you to translate raw sequencing reads into meaningful biological insights. And the best part, all the tools are accessible via Galaxy with no coding required. So now that you have seen how it works, you can try it with your own data or explore public datasets on Galaxy. So yeah, every microbiome has a story, and now you have the tools to read it. Thank you for watching, and feel completely free to reach out with any questions or feedback. And yeah, that's it.